So tell me this, what is next-gen sequencing? We hear about it so much. What is this NGS or next generation sequencing? What's the next generation about it?
So, well, I remember in 2002, I think I was a first year fellow back then, watching an announcement by Bill Clinton, Francis Collins, who still is the head of the NIH, and Craig Venter, and they announced that they had sequenced, whole genome sequenced, a human genome.
That means they took the human. What does that mean? The whole genome sequencing.
Right. So they took all of a person's genes and sequenced every single genetic codon in that person's gene. Now,
and why is that important?
Well, I think
in the
that's
context of disease.
Right, in the context of disease, because to a large extent, most of our diseases, and particularly cancer, is a genetic disease, right? Genetic alterations that lead to altered proteins, that lead to altered cells, et cetera. And I remember that there was a commentary about how much that cost. That commentary was that it cost a mere $2 billion to do so.
$2 billion with a B?
$2 billion with B. You can get online now and get your whole genome sequence for $999, right? So, I mean, there's obviously
Technology
there's a lot. There's been an incredible transformation in the technology and the sequencing technology to sequence these genes. Next-gen sequencing is really kind of has been really the reiteration of that technology, kind of on a massive scale, right? So there are vendors like Foundation and others that can now do this on a routine basis with patients in the community, et cetera. But it's able to identify whether it's just 100, 400, 300, 500, thousands of genes, and genes that we think that are truly players in oncogenesis, and to identify if there are actionable alterations that we can use to target certain drugs like larotrectinib.
Tell me this. So, some people, when they look at the new next generation sequencing, there's something called somatic mutations, there's something called germline mutations. What is the difference? And were the NTRK fusions were all somatic or germline? What's the difference between these two different mutations?
Those are good questions. So somatic just means that the actual mutation or alteration arose in the tumor. Germline refers to that it arose in the germline of the individual,
genetic thing,
such
like
as
BRCA1, BRCA2,
correct,
sometimes
such as BRCA1, that's inheritable. To date, we do not think NTRK fusions are germline, that they emerged somatically.
Mm-hmm. In the tumor itself.
In the tumor itself. It's not clear whether this is a very early event or a much later event. But at this time, we think it is a somatic alteration that has occurred in the actual tumor itself.
But tell me, this is when we talk about next-gen sequencing, some of the ones sequence tumors, and now there's also that sequence blood. So they
Correct.
call it different ways. They call it circulating tumor DNA. People call it liquid biopsies. There are different names for it. What's the inherent difference between testing the next-gen sequencing, and they're both next-gen sequencing, right?
Correct.
Doing next-gen sequencing on tissue and doing next-gen sequencing on blood. And in your study, did you have any NTRK fusions detected in blood?
Yes. CF or circulating free DNA in the blood, I'm not an expert. One of our colleagues, Dr. Phil Yanku, really is just looking at the genetic material that is being shed by tumors into the blood, oftentimes which correlate with the somatic mutations that occur in the tumor itself. It's not 100% correlated, but we do think that as the technology is emerging that there is a higher correlation, and there are commercial vendors like Guardant that are already doing that in patients in the community. We did not do in the initial data sets, we are doing in LOXO 195, for example, looking at cfDNA, but we did not do that in the initial data set for either the Navigate study or the phase 1.
Okay, so right now all of them were done on tumor tissue, right? The ones that
Correct.
you kind of identified.